talking about transgenic plants. In the previous lecture, we talked about a transgenic plant called Bt cotton. So that Bt cotton, it has some genes which can kill the insects, because Bt cotton can produce Bt toxin, right? So that's what we studied yesterday. So in the previous lecture, we studied the production of Bt cotton. Now Bt cotton we studied under insect resistant plant. We studied this under insect resistant plant. Now, how did the cotton plant gain resistance against the insect? Means the cotton plant, this transgenic cotton plant has an ability to produce Bt toxin. The cotton plant got an ability to produce Bt toxin. Now this toxin, what it is doing? The toxin present in the plant, it is killing the insect, right? This Bt toxin, it is killing the insect, and the insect dies. And the insect dies because its midgut gets affected. Because its midguts get affected. That we saw it in the previous class. So, under insect resistance, we studied about Bt cotton. Now, in today's session, we will talk about pest resistant plant, right? We'll talk about pest resistant plant. So, under pest resistant plant, we are going to study about tobacco plant. How are we going to produce a tobacco plant which shows resistance against a nematode? Ascahelminthes, Nematihelminthes. So there comes a roundworm called nematode. Okay. So roundworms are not new for us. So many nematodes we know. Several nematodes are there. They are parasites for plants also, for animals also, and for humans also. So point by point we will go with NCERT. So the NCERT says that several nematodes. There are many nematodes. Several nematodes are parasites, means they cause diseases. Several nematodes are parasites, means they cause diseases to a wide variety of. It can cause diseases to plants. They can cause diseases to animals, including humans also. It causes diseases, including in animals, including humans also. The nematodes, the roundworms will cause diseases. Do you recollect? Tell me, what is that nematode which causes disease in humans? Ascaris lumbricoides, which causes a disease called ascariasis, right? So there are nematodes which infect plant. There are nematode which infects animals also, humans also. I took a name, Ascaris causes ascariasis. Now, since we are talking about plants, let us see what is the name of a nematode which causes infection in tobacco plant. So, now the name of the nematode is a nematode. Okay, it is Melidognae. What is it called? It's called Melidognae incognitia. Melidognae incognitia. What is this? This is the name of a nematode. This is the name of a nematode. What does it causes? It will infect the root of the tobacco plant. Means this nematode is present in the soil. This nematode is present in, excuse me, where is this nematode present? The nematode is present in soil. Since it is present in soil, it infects the roots. It infects the roots of which plant? Tobacco plant. What is the scientific name of tobacco? Tobacco scientific name is Nicotiana tabacum. Nicotiana tabacum is the scientific name of tobacco. Nematode is a common name. Its scientific name is Melidognae incognitia. So this nematode present in the soil, it will infect the roots of the tobacco plant. Then the tobacco plants, if they are infected, then the roots of infected tobacco, The roots of the infected tobacco shows outgrowths. They show, what do you mean by outgrowths? Like galls, tumors. It forms galls. It forms tumors. So then the root got infected. If the root is getting infected, can it absorb water and minerals efficiently as that of a healthy root? No. So if the water is not coming out, can the leaf do photosynthesis? Because carbon dioxide and water both are required for photosynthesis. The tobacco plant cannot do photosynthesis. Now what commercially we take is the leaf only. Tobacco leaf only contains nicotine, an alkaloid. Now leaf is not doing photosynthesis, leaf is not growing, then the farmer gets a decreased yield because of this nematode called Melidognae incognitia. So it infects the tobacco roots, it shows outgrowths like galls and tumors, and what it will do? It will cause great reduction. It will cause great reduction in the yield. The yield gets reduced, right? So now we want to produce a tobacco plant which shows resistance against this nematode. Now we came out with a new technology, a novel strategy was adapted. NCERT says a novel, okay, a novel strategy was adopted. A new strategy was adapted to prevent the infestation, to prevent the nematodes infestation. A novel strategy was adapted to prevent the infection, or NCERT says infestation. To prevent the infestation. What is that novel strategy? It is based on a concept called RNA interference. To prevent the infestation by a method. What is that method called? By a method called RNA small I. We will write. What is this RNA? RNA interference. It is called as RNA interference. Now, I started taking my class. If someone comes and interrupts the class, now then the process will not go in a flow, and I couldn't teach you this because someone interrupted my session and something else continued. So we became busy. So that's what is called interference. Interference means the process will not continue. The process stopped. Okay. So how are we going to produce a tobacco plant which shows resistance against this nematode? Means there's a new concept. We came out with a new concept, novel concept. What is that called? RNA interference. RNA interference means stopping the process of RNA. Understood? What is RNA interference? RNA interference is also called RNA silencing. RNA interference is also called RNA silencing. It is also called antisense technology. It is also called antisense technology. So these are the synonyms. Whether you go with RNA interference or you are going with RNA silencing or you are calling it as antisense technology means the same. What do you mean by silencing? Let us see. So what do you mean by silencing? Means silencing of specific mRNA. See, nematode, if it has to show infection in the plant, means nematode has to make its RNA. Nematode will make RNA, and the nematode's RNA will go bind to the ribosomes, and then the nematode produces a toxin. That toxin will affect the plant. So if we see, this is a tobacco plant, right? Here are the roots, and this is the roundworm which infected. This is a roundworm which is infected. The roundworm has infected the roots. So it went into the roots of the plant. This is the root. The worm. The roundworm went inside. The roundworm went into the roots of the plant. Then this roundworm, Melidognae incognitia, will produce its mRNA. What is this called? This worm has made its RNA. So this is the mRNA of whom? mRNA of the nematode. This is the mRNA of the nematode. This mRNA of nematode, when it goes and binds to ribosomes, When it goes and binds to ribosomes, protein synthesis will happen. When protein synthesis happens, then toxic proteins are made. So then the toxic proteins is causing tumor or it is causing galls for the plant. What are we trying to do now? So we are going to make the nematodes mRNA silent. Use it. Don't talk. Don't bind to ribosomes. Don't make the proteins. So then the plant will not get affected. So that's what we are going to do. That is why we call it as RNA silencing. Again, listen carefully. Tobacco plant, tobacco leaf, tobacco root. Now, since we told it is a soil pathogen, through the soil, the roundworm went into the root. After going into the root, the roundworm has made its RNA. The roundworm has produced its RNA. Now, the mRNA of the nematode, when it goes and binds to the ribosomes, when the translation happens, then it will produce toxic proteins. Those toxic proteins will cause galls or tumors, and then the plant is infected. What we are doing? We are making the nematodes RNA silent. We are making the nematodes RNA silent. We are not allowing it to express. We are not allowing it to express. So that is called RNA silencing. That's called RNA silencing. You went and you interfered, right? You went and you interfered with the translation mechanism. You are interfering the protein synthesis mechanism so that the toxic proteins are not made. That is why it's called RNA interference. That is why it is called RNA interference. And what do you mean by antisense technology? Usually the mRNA which is made is single stranded. It will be 5 to 3 prime. The mRNA will be in 5 to 3 prime. To this RNA, if another RNA we make, to this RNA is called sense RNA. Sense RNA means, sense means direction, polarity. The usual direction for DNA or RNA, what we know is 5 to 3. It is in the same direction, 5 to 3. So it is called sense RNA. The nematode's RNA is called sense RNA. Now, you will prepare an antisense RNA. You will prepare an antisense RNA. That antisense RNA will go and bind 3 to 5. So, you made an antisense RNA which goes and binds to the sense RNA of the nematode, and this sense RNA, antisense RNA becomes double-stranded RNA. The sense RNA and the antisense RNA becomes double-stranded RNA. Only when the RNA is single-stranded, then that mRNA can bind with the rRNA which is present in the ribosomes. Again listen, only when the mRNA is single-stranded, then only it can bind to the rRNA of the ribosomes. Now when you make the mRNA to double-stranded, then can it bind to the ribosomes? It can't bind to the ribosomes. So when it can't bind to the ribosomes, the process stops. That is why we also call it as antisense technology, children. So let us have a quick recap once again. Follow carefully. We are talking about how to generate a pest-resistant plant. Then for pest-resistant plant, we are going to talk about a pest called nematode. There are many nematodes which cause parasites for plants, for animals and for humans also. We are talking about plant pest. The name of the plant nematode is Melodognia incognitia. Where is it present? In the soil. Which part of the plant does it infect? The root it will infect. It infects the root of the tobacco plant. What is the scientific name of the tobacco plant? Nicotiana tobacum. And if the roots are infected by the nematode, so see the NCERT picture. They have given you a plant. If the root gets infected, it causes tumors, galls on the control plant. Okay. So if the root is infected with tumors and galls, can it absorb water? Can it absorb minerals? Can it supply to the plant? No, it became an infected plant. Now then the yield gets reduced, it seems. Now how you are going to generate a transgenic tobacco means we came with a new technology, novel strategy called as RNA interference, also called as RNA silencing, also called as antisense technology. RNA interference. You are interfering. You are interfering. I am teaching you the class. You are listening to the class. Someone came and they are disturbing you. They are talking something else. That is interference. So, interference. You are stopping the nematode's metabolism. You are not allowing the nematode's mRNA to make the toxic protein. That is called RNA interference. You are making the nematode's mRNA silent. That is why it is called RNA silencing. The sense strand of the nematode, for that we gave the antisense strand. It became double stranded. When it becomes double stranded, it can't bind to ribosomes. It can't bind to ribosomes. Then also translation will not happen. So that is why it's called antisense technology. Take a screenshot. We will continue. Yes, understood. Now can I erase this? So we are talking about RNA interference. Now, what is RNA interference or RNA silencing? Silencing specific nematode mRNA. We are making the nematode's mRNA silent. You are not allowing the nematode's mRNA to express and produce a toxic protein. Silencing specific nematode's mRNA expression. How are you doing that? Triggered by double-stranded RNA formation. Triggered by double-stranded RNA formation, right? Double-stranded RNA formation is called RNA interference. Is called RNA interference. I told you. So, nematoda, what is that? Mylidogne incognitia will produce its mRNA, which is 5 to 3 prime direction, only if it is single stranded, then only it can bind to ribosomes. Because ribosomes have rRNA, which is also single stranded. Single stranded RNA binds with single stranded mRNA. In this mechanism, what we are doing means, incognitia has produced mRNA. To that we introduced another RNA. If this is called sense RNA of Mylidogne incognitia, we introduced antisense RNA. Sense means which goes 5 to 3. Antisense means which runs 3 to 5. When you introduced an antisense RNA, together it will become a double-stranded RNA. If it becomes a double-stranded RNA, it cannot bind to ribosome. So then what happens? Ribosomes cannot bind. If ribosomes cannot bind, no translation, no protein synthesis. No protein synthesis. If no protein synthesis, no toxin. Toxin is not there, means plant is safe. Plant is safe. Nematode will die. Nematode is not able to express its proteins. It can't survive. Nematode will die. If it is not able to express itself in the plant, then it has to die. It will die. Okay. This is what is the technology what we are going to do. Okay, na? Understood? So, silencing specific nematodes mRNA. This is the nematodes mRNA. You are making it silent. You are not allowing its mRNA expression to happen. How are you doing? You are converting it into double-stranded RNA. To the nematodes RNA, we made another RNA and converted into double-stranded RNA. If it becomes double-stranded RNA, it can't bind to ribosomes. It can't do protein synthesis. It cannot make toxic proteins. In this case, if you see, like because RNA ribosomes have bound, protein synthesis occurs. Protein synthesis occurs. Toxic protein made. Then this toxic protein, what it will do? It will infect the plant and reduces the yield. It infects the plant and it reduces the yield. Now, we are doing this technology and we are killing the nematode, protecting the plant. What is that called? RNA interference. So, we tell that RNA interference occurs in all eukaryotic organisms as a mechanism of cellular defense. All of us will have RNA interference mechanism. All of us will have RNA interference mechanism as a mechanism of cellular defense. Defense means, so for protecting ourselves, we have this mechanism. Okay, we are using it now to prepare a tobacco plant. NCERT says RNA interference occurs in all eukaryotic organisms. RNA interference occurs in all eukaryotic organisms as a mechanism of cellular defense. As a mechanism of cellular defense. To protect the cell, RNA interference our body will do. All eukaryotes have this mechanism. Understood, Amma? Take a screenshot. We'll continue. Yeah, understood? Can I erase this now? It is a little difficult concept. I am trying to explain you line by line. We are not ignoring a single word and single sentence of NCERT. Right. Who developed this technology? That also we should know, right? The invention we should know, right? So, Andrew Fire and Craig Mallow in 1998, they developed this technology. So, Andrew Fire, Craig Mallow in 1998 have developed this technology. They have or it's already there, no? They observed this technology will be better, I think. They have observed this technology. Now, where did they observe this technology? They did not observe for the nematode what we are doing, Melidogna incognita. They have done with another nematode called Sinorhabditis elegans. What is that nematode? They observed this technology in a free living, non-pathogenic, non-disease causing non-pathogenic nematode. What is the name of that? It is called as Sinorhabditis elegans. Now, check your mind. Where did you study about Sinorhabditis elegans? Where did you study? Human genome sequencing. Before doing the human genome sequencing in molecular biology, a sentence comes that, so before doing the human genome sequencing, many non-human models were sequenced by the scientist bacterial sequencing they have done, yeast sequencing they have done, Drosophila sequencing they have done, a free living non-pathogenic nematode, Sinorhabditis elegans sequence they have done, rice sequence they have done, Arabidopsis thaliana sequence they have done, all that we studied. Are you remembering that point? So, these people, Andrew Fire and Craig Mallow, in 1998 observed this technology in a free living non-pathogenic nematode. What is it? Sinorhabditis elegans. For this observation, they were awarded Nobel Prize in the year 2006. They got Nobel Prize in 2006 because it's an important discovery which helped us to prepare transgenic plants. Now, this method is involving silencing a specific mRNA, right? So, what is this? This method involves silencing specific mRNA. What are we doing here? We are making the nematode's mRNA silent. Due to complementary, you have given another RNA, complementary RNA, made it double-stranded RNA. So, due to complementary, this is what the sentence of NCERT, double-stranded RNA. What is the technology? In this technology, we are making nematode's RNA silent by converting into double-stranded RNA, and we are preventing translation. Double-stranded RNA cannot bind to ribosome, and we are preventing protein synthesis, translation. So, what are we preventing? We are preventing translation of nematode's mRNA, translation of nematode's mRNA. This is what is called silencing. This is what is called silencing. Understood, children? Now, this is nematode's RNA. This is nematode's RNA. We are calling it as sense RNA. Now, the question which should come in our mind is, from where do you get the complementary double-stranded RNA? If it is having GG, you should get an RNA which is having CC. So, then the source of complementary RNA, what is the source? The source of, to convert the single-stranded RNA into double-stranded RNA, you need a complementary RNA. Where is the source is the question. Now, there are two sources they are telling. The first source is RNA viruses. RNA viruses which have sequences complementary. RNA viruses which have sequences complementary to nematode's RNA. Now, the viruses which have RNA as the genomes, which make DNA, are called retroviruses. Now, sometimes some RNA viruses sequences might be complementary to the nematode's RNA, or else we also know that RNA is easily mutatable. RNA changes its sequence very fast. We are seeing coronavirus, alpha virus came, delta virus came, like Omicron strain came. So, it is mutating fast, it is evolving, it is showing new shapes, new sequences. So, whenever it is mutating, it will make a new sequence. So, that new sequence might be compatible or might be complementary to the nematode's RNA. So, we have to go and search. We have to go and search. Mix and match we do, right? We take top and then we go and buy for the matching. In the same manner here also, for the nematode's RNA, we are going to check the matching RNA from the RNA viruses. That is one source. The other source is mobile genetic elements. Mobile genetic elements. Mobile genetic elements are also called transposons. Mobile genetic elements are also called transposons. What do you mean by mobile genetic elements? Earlier, we used to have land number, land phone we used to have. The phone used to ring in one room. We used to fit the phone. From there, when the ring comes, wherever the person is, used to go run, pick the phone and then talk and then end the call, right? So, if anyone wants to make a call or attend a call, they should go to the phone, right? Now, we have mobile phones. On road, we are walking, we are making call. So, in this room, we are making call. Outside, we are making call. On the terrace, we are making call. So, that's mobile concept, right? In the same manner, mobile genetic elements. Now, in between this DNA, in between the sequences, so this is the mobile gene. The green colored one is the mobile gene, which is present like this. Okay, it's present like this. It's making a combination of black, green, black. It's making a combination of black, green, black. Now, this green colored one is a mobile genetic element. It can move. It went off from here. It went off from here. Next time, it can go. It's going from here. It's moving. Jumping gene. It jumped from here and it made a combination with black, green and red. New combination you got. So, means every time the mobile genetic element moves, it makes a new combination. Then also, matching can happen. We might get a sequence which is similar to the nematode's RNA. So, there is a question like from where will you get the complementary RNA? It should be complementary to the nematode's RNA. The thing is, two sources are there. One source is the RNA viruses which are having complementary sequences to the nematode's RNA. We can search. Now, is it compulsory that we are going to get? Yeah, we might get because RNA mutates easily. Since RNA mutates faster, every time when it is changing, there is possibility to get a complementary sequence. The second possibility is mobile genetic elements, jumping genes. They are also called transposons. Every time they jump from here to another place, they make a new combination. They will not stay there, amma. From there also they will jump, go to a new place, they will make a new combination. So, the source for the complementary RNA is either RNA viruses or mobile genetic elements which replicate via RNA intermediate. Mobile genetic elements means DNA, no? But what you need is RNA, no? So, that is why from DNA you have to make an RNA, then that RNA will go and bind here. Hope you understood. What did we see on this board? We are discussing who developed this technology or who identified this technology. Andrew Fire, Craig Mallow. Which year? 1998. In which nematode? In another nematode called Cynorhabditis elegans. For this, they were awarded Nobel Prize in the year 2006. What do you mean by silencing? Nematode's mRNA is made silent by giving a complementary RNA. The nematode's mRNA is made silent by giving a complementary RNA. When it becomes complementary RNA, it becomes double-stranded RNA. If it becomes double-stranded RNA, no ribosome will bind. Ribosome cannot bind. When ribosome cannot bind, no protein synthesis. No protein synthesis. No toxin made. This is the same story I am repeating again. Take a screenshot. Yes, hope you understood. Now let us talk about the mechanism exactly. With the help of diagram, I will try to tell you how to manufacture a tobacco plant, transgenic tobacco plant. Now, if you want to transfer anything into plants, what is that method you have? Agrobacterium mediated gene transfer technology. You remember? For BT cotton also we used. Right? Amma, I am borrowing two bacteria. Both these bacteria are Agrobacterium tumefaciens. The bacteria are called Agrobacterium tumefaciens. It is called natural genetic engineer. It can easily infect dicot plants. Now, this agrobacterium has Ti plasmid. What does it has? It has Ti plasmid. We took out the Ti plasmid. We opened the Ti plasmid, and now you need to keep that mobile genetic element RNA or RNA virus RNA, right? So that we are going to keep it. So we are introducing the specific sequence. So specific DNA we made. Now this is sent back into the agrobacterium. We will send it back into the bacteria. That's what I am doing. I am sending it back into the bacteria. Now this bacteria can easily infect the plant. It can easily infect the plant. We are asking this agrobacterium now to infect. We are asking the bacteria to infect the tobacco plant. What is this? Tobacco plant. Now, bacteria infected. The bacteria infected the tobacco plant, right? So, shall I write here? Infection of bacteria. Bacteria is infecting the plant. Now, after infecting the plant, what happens? Come on, tell me. So, after infecting the plant, this DNA and this DNA is here and this DNA is here. Both the DNAs undergo transcription. The DNAs will undergo transcription and they will make RNAs, right? So, let me show you. So, one DNA, another DNA, you send it into the tobacco plant. Now, it makes one RNA, it makes another RNA. So, this strand after transcription will make one RNA, the other strand also will make other RNA. So, this RNA will be 5 to 3, the other RNA will be 3 to 5. So, inside the tobacco plant, you got a double-stranded RNA. After transcription, we got double-stranded RNA. This double-stranded RNA will come into the cytoplasm of the cell. The double-stranded RNA, the bacteria infected the plantama. After infecting the plant, this is DNA. DNA after transcription, it will make RNA. One RNA, other RNA, both are complementary to each other. You got a double-stranded RNA. Now, in the plant, we told cellular defense is there. In the plant, this double-stranded RNAs are identified. They are identified by dicer protein. They are identified by dicer protein. Now, let us see what is this dicer protein. It is double-stranded. It is double-stranded specific ribonuclease protein. It will identify double-stranded RNAs and it will break the double-stranded RNAs. Now, dicer protein will go and identify double-stranded RNAs. It will break the double-stranded RNAs. Now, what happens? The dicer protein has identified this double-stranded RNA. It is breaking it into small pieces. It has broken it. If you make it big, then you got two pieces, this much and this much. It's breaking into two small RNAs. These two small RNAs are called siRNAs. These two small RNAs are called small interfering RNAs. What are they called? Small interfering RNAs. So, this small interfering RNAs, if you see, in this small interfering RNA, one is going 5 to 3, the other one is going 3 to 5, correct? One strand is 5 to 3, the other strand is 3 to 5. Now, the one which is 5 to 3 is called passenger RNA. The one which is 3 to 5 is called guide RNA. This is passenger RNA then, and this is guide RNA then. The passenger RNA is broken off. See, passenger RNA is broken off. Guide RNA will be intact. The guide RNA will be intact. Now, this guide RNA is now identified by one more complex, which is called RNA Induced Silencing Complex. There's one more complex. What is this called? RNA Induced Silencing Complex. What is it called? I'll try to show. This is called RNA Induced Silencing Complex. In that, it has a protein here. It has one protein there. It has another proteins here. This protein is called as a granulate protein. This protein, which is very important, is called a granulate protein, and these two are the other proteins. Now, what this RNA induced silencing complex will do? RNA induced silencing complex will take the guide RNA inside. It is taking the guide RNA inside. Yes, this is the guide RNA. It took the guide RNA, which is 3 to 5. Now, this RNA induced silencing complex and the guide RNA are in search of nematodes RNA. These two are in search of, nematodes mRNA. Now, if this plant, which is having all this is inside, if this plant, because agrobacterium has sent the genes, right, means it is a transgenic tobacco. It is a transgenic tobacco plant because agrobacterium has transferred the genes. These genes made the double-stranded RNA. The double-stranded RNA is identified by the dicer protein. Dicer protein diced it off into siRNAs. In siRNA, one strand of RNA, which is 5 to 3, is called as passenger RNA. The other strand of RNA, which is 3 to 5, is called guide RNA. Passenger RNA is broken off. Guide RNA will be intact. Guide RNA will take the help of RICS, RNA Induced Silencing Complex. Both together are going in search of nematodes RNA. If at all such transgenic tobacco is infected by a roundworm, if it is infected by this is a roundworm. The roundworm, after infecting the plant, it will make an mRNA. The roundworm has made its mRNA. Now, this mRNA which is single stranded is being found by this, right? So, this mRNA, wait, shall I change the color? Now, this is roundworm's mRNA. Now, this RNA induced silencing complex with the guide RNA, if it finds the nematodes RNA, if it finds the nematode RNA, it takes the nematodes RNA. It takes the nematodes RNA, and what it will do? It will bring a scissor, cut it off. It will bring a scissor and it cuts off the nematodes RNA. So, the nematodes RNA broken, amma. The nematodes RNA broken, the roundworm's RNA is broken into pieces. That means nematodes RNA is not free. First it made into double-stranded RNA. It converted into double-stranded RNA. So, and then finally it will break it also. When it cleaves off, then if this RNA is not there, nematode will survive or nematode will die. Nematode will die. So, this is the technology about. Understood? So, the guide RNA is further going and binding to RNA induced silencing complex. This is the RNA induced silencing complex which has a granulate protein and it has two more proteins which take the guide RNA, and both of them are going in search of nematodes RNA. If at all such a plant is infected with a roundworm, if it makes the roundworm makes the RNA, that RNA will go and bind to this, and this RISC and the guide RNA will cleave it off. If the nematodes RNA is cleaved off, nematode cannot survive, right? If nematode is not surviving, it means it is dead. Means the plant is safe. The transgenic plant is safe. Infection or no infection? No infection also. Okay, children. All this is, what is this called? This is called RNA interference. It is also called RNA silencing. Take a screenshot. Right? Understood? Now, this is our lectures about biotechnology and its applications. Now, in the next session, we'll start a new chapter called Microbes in Human Welfare. Hope the content is informative. So, any suggestions? Yeah, you can post them in the comment box. You're welcome. If you like the content, like, share and subscribe to my channel. Thank you.